A patient dies from a myocardial infarction. Another patient's immune system eliminates a virus-infected hepatocyte. Both cells are dead, but on the step 1 exam, only one triggers inflammation. Do you know which one and why? That answer tells you everything about the difference between necrosis and apoptosis. Let's build it. Here's the concept that anchors everything else. Necrosis is uncontrolled cell death from external injury. The cell is overwhelmed, its membrane ruptures, and its contents spill into the surrounding tissue. That spillage triggers inflammation, every time. Apoptosis is programmed cell death. The cell dismantles itself from within, packages its contents into neat membrane-bound fragments called apoptotic bodies, and those fragments are quietly phagocytosed. No spillage, no inflammation. That single difference, inflammation yes versus inflammation no, is the most tested concept in this entire topic. Now let's go deeper into necrosis. There are 5 types you need to own for step 1. And here's the key insight that ties them together. The type of necrosis you see tells you exactly what happened to the tissue and what caused it. Think of necrosis like a crime scene. The pattern of death is the evidence. Type 1 is coagulative necrosis. This is the default. When any solid organ loses its blood supply, proteins denature and coagulate. The cell's structural outline is preserved, so under the microscope you can still see ghost-like cell borders, but the nucleus is gone. The classic example is myocardial infarction. Heart, kidney, and spleen infarcts are all coagulative. There is one critical exception that step 1 loves: the brain. Brain infarction does not cause coagulative necrosis. The brain is rich in lipids and hydrolytic enzymes. When ischemia hits, those enzymes digest tissue before proteins can coagulate. The tissue liquefies instead, which brings us to type 2. Type 2 is liquefactive necrosis. Here, the tissue completely dissolves into a liquid mass. There are only 2 causes you need to remember: brain infarction and pyogenic bacterial infections. Both flood tissue with hydrolytic enzymes, either from injured brain cells or from neutrophils recruited by bacteria. The product of liquefactive necrosis in infection is pus. An abscess is localized liquefactive necrosis. If you see pus, abscess, or a fluid-filled cavity, think liquefactive. Type 3 is caseous necrosis. This looks like cottage cheese, soft, white, and friable. It is pathognomonic for tuberculosis. Mycobacterial cell wall lipids resist digestion by macrophages. The immune system walls the area off into a granuloma, and the center undergoes a hybrid of coagulative and liquefactive necrosis. No cellular architecture remains, just amorphous debris surrounded by epithelioid macrophages and Langhans giant cells. Certain fungal infections like histoplasma and coccidioides can also cause caseous necrosis. Type 4 is fat necrosis. There are 2 settings: acute pancreatitis and breast trauma. In pancreatitis, activated pancreatic lipases escape the gland and digest peripancreatic fat. This releases free fatty acids, which bind calcium in a process called saponification. The result is chalky white deposits. This is also why acute pancreatitis can cause hypocalcemia. In breast trauma, fat cells rupture and undergo the same saponification process. Type 5 is fibrinoid necrosis. This only happens in blood vessel walls. Immune complexes deposit in the vessel wall, activate complement, recruit neutrophils, and neutrophil enzymes destroy the wall. On histology, the vessel wall looks bright pink and fibrin-like. Classic associations include polyarteritis nodosa, malignant hypertension, rheumatic fever, and lupus vasculitis. 2 bonus patterns show up in vignettes. Dry gangrene is coagulative necrosis from ischemia without infection, like a mummified black diabetic toe. Wet gangrene is coagulative plus liquefactive necrosis due to bacterial superinfection on ischemic tissue. One more necrosis detail step 1 loves: the nuclear death sequence. In necrosis, the nucleus dies in a predictable order. Pyknosis, the nucleus shrinks and darkens. Karyorrhexis, it fragments. Karyolysis, it dissolves. PKL, always in that order. Now shift to the right side of the screen, apoptosis. 2 pathways trigger apoptosis, and both converge on the same executioner, caspases, proteases that systematically dismantle the cell. The intrinsic pathway is mitochondrial-driven. Internal signals like DNA damage, oxidative stress, or growth factor withdrawal shift the balance between pro-apoptotic proteins, such as BAX, and anti-apoptotic proteins, such as BCL2. When BAX wins, it punches holes in the outer mitochondrial membrane. Cytochrome C leaks out, binds APAF1, forms the apoptosisome, and activates caspase 9. That triggers the downstream caspase cascade, and the cell undergoes apoptosis. Here is your step 1 pearl. P53 promotes apoptosis by suppressing BCL2. Massive DNA damage activates P53. BCL2 is downregulated and apoptosis proceeds. Loss of P53 is dangerous because cells with catastrophic DNA damage survive when they should die. The flip side is BCL2 overexpression. Excess BCL2 blocks apoptosis. This is exactly what happens in follicular lymphoma. A T1418 translocation places BCL2 under immunoglobulin promoter control. BCL2 is overexpressed, and B cells that should die keep surviving. The extrinsic pathway is death receptor-driven. External signals activate receptors on the cell surface. The two classic examples are Fas ligand binding the Fas receptor and TNF-alpha binding TNFR1. These receptors recruit an adaptor protein called FADD, which activates caspase 8 directly, bypassing the mitochondria. Caspase 8 then triggers the same executioner caspase cascade. The classic cellular example is cytotoxic T cells killing virus-infected cells. The T cell expresses Fas ligand. It binds Fas on the target cell, and apoptosis occurs. Clean removal, no inflammation. That is exactly what you want when eliminating infected cells in healthy tissue. The single most important apoptosis lab finding is DNA laddering. Caspase-activated endonucleases cleave DNA at regular intervals between nucleosomes, producing fragments of about 180 base pairs and multiples of that. On a gel, this looks like a ladder. Necrosis produces random DNA smearing. If a vignette mentions DNA laddering, the answer is apoptosis. Now let's hit 3 vignette traps. These separate students who know facts from students who get questions right. Trap 1 is the inflammation question. A patient has viral hepatitis. A liver biopsy shows scattered dead hepatocytes. What type of cell death is this, and does it cause inflammation? The answer is apoptosis. Virus-infected hepatocytes are killed by cytotoxic T cells via Fas ligand pathway. This does not cause significant inflammation. The biopsy shows acidophil bodies, also called Councilman bodies, which are apoptotic bodies, not necrotic debris. Trap 2 is the brain infarct exception. A patient has an MCA stroke. 2 weeks later, brain tissue shows a fluid-filled kystic cavity. What type of necrosis is this? Liquefactive. The brain is the one solid organ exception to coagulative necrosis. The histologic sequence is red neurons of 12 to 24 hours, neutrophils at 1 to 3 days, macrophages at 1 to 3 weeks, and finally a kystic cavity. That cavity is the end stage of liquefactive necrosis. Trap 3 is the pancreatic calcium question. A patient with acute pancreatitis has hypocalcemia. Why? Fat necrosis. Activated lipases digest fat. Free fatty acids bind calcium through saponification, and serum calcium drops. Students who see hypocalcemia reflexively think parathyroid hormone or vitamin D. The vignette gives you pancreatitis. The mechanism is fat necrosis. Let's close with what you now own. First, necrosis is uncontrolled. The membrane ruptures, contents spill, and inflammation always follows. 5 types matter: coagulative everywhere except brain, liquefactive in brain and pyogenic infections, caseous in tuberculosis, fat necrosis in pancreatitis, fibrinoid in vessel walls. Nuclear sequence is Pyknosis, Karyorrhexis, Karyolysis, PKL, always in that order. Second, apoptosis is programmed and silent, no inflammation. The intrinsic pathway runs through mitochondria, cytochrome C, BCL2, P53, and caspase 9. The extrinsic pathway runs through Fas or TNFR1, FADD, and caspase 8. DNA laddering means apoptosis. Third, the traps. Viral hepatitis hepatocyte death is apoptosis, not necrosis. Brain infarction is liquefactive, not coagulative. Pancreatitis hypocalcemia is fat necrosis from saponification, not a calcium regulation disorder. Subscribe to Step Genie,
Every video is built exactly like this one. Drop your hardest step 1 topic in the comments. Next week, inflammation, acute versus chronic, the cellular players, and why neutrophils show up first. See you there.